For millennia, humanity has cast its gaze skyward, captivated by the twinkling tapestry of stars, the silent ballet of planets, and the distant, enigmatic glow of nebulae. We build colossal telescopes that pierce the veil of light years, launch probes that traverse the cold void between worlds, and dream of colonizing celestial bodies far beyond our reach. This profound fascination with the cosmic unknown drives some of our most ambitious scientific endeavors, pushing the boundaries of engineering and human imagination. We yearn to understand the universe's grand design, to find signs of life beyond Earth, to unravel the secrets held in the stardust of ancient galaxies. Yet, in our spectacular pursuit of the distant and the divine, we often overlook a universe of equal, if not greater, complexity and mystery that lies not light years away, but mere inches beneath our feet. What we casually refer to as dirt is, in fact, a vibrant, intricate, and profoundly influential living system. It is the very foundation of nearly all terrestrial life, a silent, tireless engine driving countless planetary processes. This ubiquitous brown layer, which we walk upon, build upon, and often dismiss as inert matter, is anything but. It is a frontier as vast and unexplored in its own way as the deepest ocean trench or the most distant exoplanet. The paradox is striking. We spend billions exploring the potential for life on Mars, while the teeming, undeniable reality of life in the soil beneath our gardens, our farms, and our wild forests remains largely unseen, unappreciated, and fundamentally misunderstood by most. Imagine a single scoop of earth, perhaps just a handful of what you might find in your backyard. To the naked eye, it might appear homogeneous, a simple mixture of gritty particles and dark clumps. Perhaps a stray root, a small pebble, or a deceased leaf fragment is visible. But this seemingly unremarkable sample holds a population far denser and more diverse than any human city. Within that small volume, an entire universe unfolds, a hidden metropolis bustling with billions of inhabitants. It is a world where microscopic organisms engage in a ceaseless dance of decomposition, transformation, and creation, shaping the very fabric of our planet in ways we are only just beginning to fully comprehend. This isn't just a medium for plants to root in. It is an active participant, a dynamic entity that breathes, filters, recycles, and communicates. Far from being a mere substrate, a simple, inert base material, soil is a living, evolving matrix, constantly engaged in a complex interplay of physical, chemical, and biological forces. Its composition is never static. It is perpetually being built up and broken down, enriched and depleted, shaped by the relentless work of its countless residents and the forces of nature. The perception of soil as just dirt implies a lack of vitality, a passive substance awaiting external influence. But this couldn't be further from the truth. Soil is intensely active, a vast, slow-motion factory operating 24 hours a day, 7 days a week, without breaks or holidays. To truly appreciate soil, we must peel back the layers of our preconceptions. We must look beyond the surface, past the dust that settles on our shoes, or the mud that clings to our tires. We must acknowledge that this dark, earthy substance is a stratified masterpiece, a geological and biological archive, with each horizon or layer telling a story of countless millennia. From the crumbly topsoil, rich in organic matter, to the deeper, more mineral-dominated layers, down to the bedrock from which it all ultimately originated. Soil represents a continuum of life and geological process. Its texture, its color, its very smell, that distinct earthy aroma often associated with rain or freshly turned earth. are all indicators of its profound biological activity, a telltale sign of the intricate chemistry and microbiology at play. The hidden world of soil is a realm of darkness and dampness, a place where sunlight rarely penetrates, yet where life thrives in astonishing abundance. It is a world governed by cycles of decay and renewal, where the discarded remnants of macroscopic life, fallen leaves, dead wood, animal remains, are meticulously broken down and reintegrated into the system. This constant recycling of nutrients is not a simple chemical reaction. It is orchestrated by an army of microscopic engineers, each playing a specific role in transforming complex organic molecules into simpler forms that can be utilized by plants and other organisms. Without this tireless process, the surface of our planet would be buried under an ever-growing accumulation of dead organic matter, and the essential nutrients required for new life would remain locked away, inaccessible. The very air we breathe and the water we drink are intimately connected to the health and function of soil. It acts as a vast, living filter, purifying water as it percolates downwards, removing contaminants and regulating its flow into groundwater reserves and rivers. It participates in the global carbon cycle on a scale comparable to, or even exceeding, that of the atmosphere and oceans, acting as a colossal reservoir for carbon, a key element in regulating Earth's climate. Every breath of air, every sip of water, every bite of food we consume traces its origins back to this often ignored realm beneath our feet. Its silent work underpins the stability of ecosystems, the productivity of agriculture, and ultimately, the survival of humanity itself. Consider the sheer scale of this overlooked importance. Soil is the foundation for virtually all terrestrial ecosystems, from the towering redwoods of California to the sprawling grasslands of the Serengeti, from the dense rainforests of the Amazon to the seemingly barren tundras of the Arctic. Life on land is inextricably linked to the quality and depth of the soil. It provides physical support for plants, anchoring them against wind and weather. It stores and supplies the water and nutrients essential for their growth. In turn, these plants form the base of the food web, feeding herbivores, which then feed carnivores, creating the intricate tapestry of biodiversity that defines our planet. Without healthy soil, this entire edifice crumbles. The paradox, then, is not merely intellectual. It has profound practical implications. How can something so fundamental, so utterly indispensable, be so routinely overlooked, mistreated and degraded? Our casual perception of dirt has led to practices that deplete its vitality, erode its structure, and compromise its ability to sustain life. We have treated it as an expendable resource, a passive medium to be exploited, rather than a living system to be nurtured and understood. Unveiling the secret life of soil is not just a scientific curiosity, it is an urgent necessity for our collective future. It is an invitation to look down, to truly see and appreciate the vibrant, complex, and vital world hidden just beneath the surface. A world whose health determines our own. We are about to embark on a journey into this incredible, invisible frontier, to explore the quiet, invisible engine that powers our planet. We often marvel at the vastness of the cosmos, pondering the existence of life on distant planets, yet perhaps the most incredible, densely populated, and utterly vital metropolis lies hidden beneath our very feet. Take a moment to imagine scooping up a single handful of ordinary soil. What you hold is not inert dirt, but an entire universe, teeming with an astonishing array of life forms, each playing a crucial, intricate role in the planet's silent, continuous operation. This unassuming clump of earth harbors more living organisms than there are people on the entire planet. Perhaps even more species than exist in the sprawling Amazon rainforest. It is a microscopic city, vibrant and ceaselessly active, where billions of inhabitants go about their daily lives unseen and largely unappreciated by the macroscopic world above. At the very foundation of this subterranean metropolis are the microorganisms, the tiny, single-celled architects that drive the vast majority of soil processes. Among them, bacteria are the undisputed champions of sheer numbers and diversity. A single gram of healthy soil, roughly the weight of a paperclip, can contain anywhere from 100 million to 1 billion individual bacterial cells, representing thousands of distinct species. These are prokaryotes, meaning their cells lack a nucleus, but their simplicity belies their profound capabilities. They exist in an incredible variety of forms and metabolisms, clinging to the surfaces of mineral particles, thriving in thin films of water within soil pores, and colonizing the decaying remains of plants and animals. Their primary role is that of relentless decomposers, breaking down complex organic compounds into simpler forms, making nutrients available for plants and other organisms. Without them, the earth would be buried under layers of undecayed organic matter, and the cycles of life would grind to a halt. Closely related, yet distinct in their evolutionary lineage, are archaea. Once mistaken for bacteria, these ancient microorganisms are now recognized as a separate domain of life. While often associated with extreme environments like hot springs or deep-sea vents, archaea are also abundant in soil, contributing significantly to the global cycles of nitrogen and carbon. Some archaea play a part in converting ammonia into nitrites, a critical step in the nitrogen cycle, while others are involved in the production or consumption of methane, a potent greenhouse gas. Their presence highlights the incredible biochemical diversity that thrives just inches below the surface, performing vital planetary functions that underpin the very air we breathe and the food we eat. Beyond these single-celled powerhouses, the soil metropolis is interwoven with the intricate structures of fungi. Unlike bacteria and archaea, fungi are eukaryotes, possessing a nucleus within their cells, and often grow as vast networks of thread-like structures called hyphae. If you were to unspool all the fungal hyphae from just 1 gram of forest soil, it could stretch for several kilometers. These incredible networks are the primary decomposers of tough, woody materials like cellulose and lignin, which bacteria find difficult to break down. Fungi act as the master recyclers, transforming dead plant material into nutrient-rich organic matter. Their hyphae also act as natural glues, binding individual soil particles together to form stable aggregates, which are crucial for creating healthy soil structure. This structure, much like the foundation of a building, dictates how well water infiltrates, how air circulates, and how easily plant roots can penetrate the soil to access nutrients. Completing the cast of microscopic characters are the protozoa. These are single-celled eukaryotes, often larger than bacteria, and they come in diverse forms. Amoebas that ooze through water films, ciliates propelled by tiny hair-like structures, and flagellates that whip their tails to move. Protozoa are primarily predators, grazing on bacteria and other smaller microorganisms. This seemingly simple act of consumption has a profound impact on nutrient cycling. When protozoa eat bacteria, they release excess nutrients, particularly nitrogen, that were locked up within the bacterial cells. This process, known as mineralization, makes these vital nutrients available for uptake by plants. effectively acting as a nutrient release valve in the soil ecosystem. Their presence is a strong indicator of an active and healthy microbial food web, ensuring that essential elements are continuously recycled and made accessible. Together, these microscopic inhabitants are the primary drivers of decomposition, relentlessly breaking down the remains of dead plants and animals. Imagine a fallen leaf. It doesn't simply vanish. Instead, it becomes a banquet for an unseen army. Bacteria and fungi begin the initial assault, secreting enzymes that dissolve the leaf's complex molecules. Over time, the leaf is transformed from solid organic matter into simpler compounds, some of which are absorbed by the microbes, while others are released into the soil or incorporated into a stable, dark material called humus. This humus is not just decomposed organic matter, it's a reservoir of carbon and nutrients, improving soil structure and water retention, and acting as a slow-release fertilizer for plants. This constant, quiet work ensures that the earth never runs out of the building blocks of life. This decomposition is intricately linked to nutrient cycling, a series of processes that transform essential elements like nitrogen, phosphorus, and carbon into forms that plants can use. Consider the nitrogen cycle. The air we breathe is about 78% nitrogen gas, but plants cannot directly utilize it in this form. Specialized bacteria, known as nitrogen fixers, convert atmospheric nitrogen into ammonia, a usable compound. Other bacteria then transform this ammonia into nitrites and nitrates through a process called nitrification. Nitrates are the primary form of nitrogen absorbed by plants. When plants and animals die, decomposers return this nitrogen to the soil, and still other bacteria, called denitrifiers, convert some of it back into nitrogen gas, completing the cycle and releasing it into the atmosphere. This complex dance of microbial activity ensures a continuous flow of nutrients, making life on land possible. Every nutrient that nourishes a plant, an animal, or ultimately us, has likely passed through the metabolic machinery of countless microorganisms. Beyond the microbial realm, the soil metropolis also hosts a diverse array of larger, though still often hidden, inhabitants, the invertebrates. These creatures are the macro and mesofauna, providing the crucial physical labor that complements the biochemical work of the microbes. Among the most important are nematodes, tiny, unsegmented roundworms, often no more than a millimeter long. While some nematodes are parasitic and can harm plants, the vast majority are beneficial, performing vital roles in the soil food web. Bacterial-feeding nematodes graze on bacteria, fungal-feeding nematodes consume fungi, and predatory nematodes hunt other nematodes and protozoa. Like protozoa, when these nematodes consume microorganisms, they excrete nutrients in a plant-available form, accelerating nutrient cycling. Their sheer abundance, often numbering in the thousands per handful of soil, underscores their collective impact. Perhaps the most iconic engineers of the soil are earthworms. These remarkable creatures, with their segmented bodies and tireless burrowing, are often called the intestines of the earth. As they tunnel through the soil, they create an intricate network of channels, some reaching depths of several meters. These burrows serve as natural conduits for air and water, vastly improving soil aeration and drainage. Without earthworms, water would struggle to penetrate compacted soils, leading to increased runoff and erosion. As they ingest soil and organic matter, they grind it down, mix it, and then excrete it as nutrient-rich casts, which are essentially miniature, perfectly formed fertilizer pellets. These casts are stable soil aggregates, further enhancing soil structure. Different types of earthworms specialize in different tasks. Some live near the surface, feeding on leaf litter. Others create horizontal burrows, mixing topsoil. And still others, like the common nightcrawler, dig deep, permanent vertical shafts, pulling organic matter down from the surface into the deeper layers of the soil. Their combined activity is an ongoing vital process of physical breakdown and redistribution, making the soil a more hospitable and productive environment for all its inhabitants. Finally, we find a fascinating array of arthropods, a broad group that includes insects, mites, springtails, collembola, millipedes, and centipedes. These creatures vary greatly in size, from the barely visible mites to the more obvious millipedes. Their primary contribution is the physical fragmentation of organic matter. Imagine a fallen log or a thick layer of leaf litter, while fungi and bacteria will eventually break it down. Arthropods act as the initial processors, chewing, shredding, and tearing larger pieces into smaller fragments. This increases the surface area available for microbial decomposition, accelerating the entire recycling process. Springtails, for instance, are tiny, jumping insects that graze on fungi and decaying plant material, helping to spread fungal spores and break down detritus. Mites, another incredibly diverse group, can be decomposers, predators, or fungal feeders, further complicating and enriching the soil food web. Millipedes are diligent detritivores, consuming decaying plant matter, while centipedes are swift predators, helping to regulate populations of other soil organisms. In essence, a handful of soil is not merely a collection of particles. It is a bustling, interdependent community, a true metropolis of life. From the billions of bacteria and archaea driving biochemical transformations, to the extensive fungal networks recycling tough organic matter, to the protozoa releasing vital nutrients, and the earthworms and arthropods physically engineering and aerating the soil. Every inhabitant plays an indispensable role. This complex web of interactions ensures the continuous flow of energy and nutrients, forming the very foundation upon which all terrestrial ecosystems are built. This silent, invisible engine, constantly working beneath our feet, is far more alive and dynamic than we often imagine, a testament to the planet's intricate and self-sustaining systems. After exploring the teeming microscopic metropolis within a handful of soil, our journey now takes us even deeper, into a realm that blurs the lines between individual organisms and a collective intelligence. We move from the bustling streets of bacteria and archaea to the vast, intricate highways of another essential life form, fungi. When we think of fungi, our minds often conjure images of mushrooms sprouting after a rain, their caps a brief visible declaration of existence. Yet, these familiar fruiting bodies are merely the tip of a colossal hidden iceberg, the ephemeral flowers of an organism that lives almost entirely beneath our feet, weaving through the soil in an astonishing network of threads. These threads are called hyphae, and collectively they form a mycelium, the true body of a fungus. Imagine an intricate gossamer web, finer than human hair, stretching for miles within just a cubic foot of soil. These hyphae are not merely passive structures. They are actively growing, sensing, and exploring the subterranean environment, seeking out nutrients and forging connections. A single gram of forest soil, an amount equivalent to a paperclip, can contain several kilometers of these fungal threads. They are the silent, tireless miners of the soil, extending far beyond the reach of plant roots, navigating the labyrinthine spaces between mineral particles and organic matter. What makes these fungal networks truly extraordinary is their capacity to connect plants, not just individual plants of the same species, but often diverse species across an entire ecosystem. This phenomenon has earned a fitting, almost poetic nickname, the Wood Wide Web. Coined by scientists, most notably Dr. Suzanne Simard from the University of British Columbia, this term vividly describes a subterranean communication and resource-sharing system, a biological internet that underpins the health and resilience of nearly all terrestrial plant life. Imagine a forest where every tree, every shrub, and even many smaller herbaceous plants are subtly linked, exchanging information and vital resources through this living underground matrix. At the heart of the Wood Wide Web are symbiotic relationships, particularly those known as mycorrhizal associations. The word mycorrhiza itself is derived from Greek, meaning fungus root, a direct and accurate description of this ancient partnership. For millions of years, fungi and plants have co-evolved, forming a mutually beneficial relationship where each partner provides something essential to the other. Neither could thrive, or perhaps even survive, in their current forms without this intricate dance of exchange. Let's delve into what each partner gains from this remarkable alliance. For the plant, the benefits are profound and multifaceted. Plant roots, even with their fine root hairs, have a limited surface area and can only explore a finite volume of soil. Fungal hyphae, however, are vastly more extensive and significantly thinner, allowing them to penetrate tiny pores and access nutrient reserves that would otherwise be beyond the plant's reach. Think of it like a plant having a highly specialized, infinitely branching extension cord that can plug into every available outlet in its vicinity. These fungal partners are particularly adept at acquiring phosphorus and nitrogen, two critical macronutrients that are often scarce or immobile in soil. Phosphorus, for instance, tends to bind tightly to soil particles, making it difficult for roots to absorb. The fungi release enzymes and organic acids that can unlock these bound nutrients, bringing them directly to the plant's root cells. Beyond nutrient acquisition, mycorrhizal fungi also enhance a plant's access to water. In dry conditions, their vast networks can tap into minute pockets of moisture within the soil matrix, effectively extending the plant's drought tolerance. Some studies suggest that plants connected to extensive fungal networks are more resilient to water stress, drawing on a shared reservoir of moisture when individual plants are struggling. Furthermore, these fungi can offer a degree of protection against soil-borne pathogens and diseases. They can form a physical barrier around the root, or even produce antimicrobial compounds that deter harmful microorganisms, acting as a kind of immune system for the root zone. In return for these invaluable services, the fungi receive a steady supply of carbohydrates, sugars produced by the plant through photosynthesis. Fungi, unlike plants, cannot harness sunlight to create their own food, making them reliant on other organisms for energy. The plant essentially feeds its fungal partner, providing the fuel necessary for the mycelium to grow, explore, and continue its beneficial work. It's a perfect example of biological reciprocity, a finely tuned economic system operating beneath the surface. There are two main types of mycorrhizal associations that dominate terrestrial ecosystems. The first are ectomycorrhizal fungi, often associated with forest trees like pines, oaks, birches, and firs. These fungi typically form a dense sheath, almost a glove, around the outside of the root tips. From this sheath, Hyphae extend deeply into the soil and also grow between seed, the root cells, forming what is known as the Hartig net, where the crucial nutrient exchange occurs. The other major group is arbuscular mycorrhizal fungi, often abbreviated as AMF, which are far more widespread, associating with about 80% of all plant species, including most agricultural crops, grasses, and many herbaceous plants. AMF fungi don't form an external sheath. Instead, their hyphae penetrate into the root cells themselves, forming intricate tree-like structures called arbuscules, where the nutrient exchange takes place, and sometimes storage vesicles. This direct cellular integration highlights the deep evolutionary partnership between these organisms. The Wood Wide Web is not just about nutrient transfer, it's also a sophisticated communication network. Imagine a forest where a single tree is under attack by an insect pest, such as aphids. This tree can release chemical signals, often volatile organic compounds, into the soil. Through its mycorrhizal connections, these warning signals can travel through the fungal network to neighboring trees. These recipient trees, even if they haven't yet been attacked, can then prime their defenses, ramping up production of defensive chemicals or toxins that make them less palatable to the incoming pests. This early warning system can provide a significant survival advantage, allowing the entire forest community to respond more quickly and effectively to threats. Dr. Suzanne Simard's pioneering research in the forests of British Columbia provided some of the most compelling evidence for this underground communication. Using isotopic tracers, she demonstrated that carbon, fixed by photosynthesis in one tree species, like a paper birch, could be transferred through mycorrhizal networks to another species, such as a Douglas fir, and vice versa. This was particularly evident when one species was shaded or stressed, suggesting that the network acts as a support system, allowing resources to be redistributed to where they are most needed. She observed that older, larger mother trees could even send carbon and nutrients to young seedlings, helping them establish and thrive in challenging conditions, effectively nurturing the next generation of the forest. This isn't just a simple transfer. It's a dynamic, responsive allocation of resources, a testament to the network's complexity. Consider the practical implications of such a system. In a dense forest, light is a precious commodity. A young sapling, struggling in the deep shade of a mature canopy, might receive a critical lifeline of sugars from its larger, sun-drenched neighbors through the fungal network. This shared economy helps maintain the overall health and biodiversity of the ecosystem, ensuring that resources are distributed efficiently rather than being hoarded by the most dominant individuals. It's a cooperative model that challenges the traditional view of plants as purely competitive entities. This biological internet also facilitates the movement of water. During periods of drought, an older, deep-rooted tree might have access to deeper water sources than a shallower-rooted younger plant. Through the fungal network, this water can potentially be shared, mitigating the effects of localized water stress and enhancing the resilience of the entire plant community. While the exact mechanics and extent of water transfer are still subjects of ongoing research, the principle of shared resources through the mycelial network is increasingly accepted. However, it's important to remember that this system isn't always a utopian model of altruism. Like any complex network, it has its nuances and even its dark side. While cooperation is prevalent, competition still exists. Some plants might be better negotiators in the fungal exchange, or some fungi might preferentially allocate resources. There can be instances of cheating, where one partner takes more than it gives. Though such imbalances are generally self-correcting over ecological timescales. The Wood Wide Web is not a single unified entity, but a dynamic, constantly shifting tapestry of countless individual interactions, each influenced by local conditions, species present, and environmental pressures. The scientific community continues to unravel the mysteries of this underground internet. Researchers are exploring the specific chemical signals involved in plant-to-plant communication, the precise mechanisms of nutrient and water transfer, and how these networks respond to environmental changes like climate shifts, pollution, or agricultural practices. The understanding of these fungal networks has profound implications for how we manage our forests, design agricultural systems, and approach conservation. Conventional farming practices such as intensive tilling, heavy use of synthetic fertilizers, and monocropping can severely disrupt or destroy these delicate mycelial networks. When the soil is turned over repeatedly, the fine fungal threads are fragmented, breaking the essential connections between plants. This forces plants to rely solely on their own roots for nutrient uptake, often leading to increased dependence on external fertilizers and reduced resilience to stress. In contrast, regenerative agricultural practices, which prioritize minimal soil disturbance, cover cropping, and diverse crop rotations, actively work to preserve and rebuild these vital fungal networks. By fostering healthy soil microbiology, farmers can reduce their reliance on synthetic inputs, improve soil structure, enhance water retention, and increase crop yields in a sustainable manner. The health of these invisible networks directly translates into the health of our food systems and the broader environment. So when we consider soil, it's not merely a medium for roots or a static collection of dirt particles. It is a living, breathing, communicating entity, a vast subterranean brain that connects the life above ground in ways we are only just beginning to comprehend. The mycelial networks, the Wood Wide Web, stand as a testament to the profound interconnectedness of life on Earth, operating silently beneath our feet, orchestrating a complex ballet of resource exchange and communication that is fundamental to the survival of nearly every terrestrial ecosystem. It reminds us that some of the most sophisticated systems on our planet are often the most overlooked. Beyond the bustling, unseen cities of microorganisms and the intricate, whispering networks of fungi beneath our feet, soil operates on an even grander, more planetary scale. It is not merely a foundation, but a dynamic, living organ of Earth itself, performing critical functions that ripple through every global cycle. Imagine our planet as a colossal living being. Then soil is its breathing skin, constantly inhaling and exhaling, regulating temperature, filtering vital fluids, and providing the very sustenance for its surface life. This silent, tireless work shapes the atmosphere we breathe, the water we drink, and the food we eat, yet often goes unnoticed. One of soil's most profound planetary roles lies within the global carbon cycle. Carbon, the essential building block of life, moves continuously between the atmosphere, oceans, rocks, and living organisms. Soil acts as an immense and often overlooked reservoir in this grand dance. When we speak of soil breathing, we are referring to the constant exchange of gases, primarily carbon dioxide and oxygen, between the soil and the atmosphere above it. This exchange is driven by the tireless activity of countless soil organisms and the decomposition of organic matter. Let's delve deeper into how soil manages carbon. Plants, through photosynthesis, draw carbon dioxide from the atmosphere, converting it into sugars and structural biomass. a significant portion of this carbon is then transferred to the soil. Some enters directly as fallen leaves, dead roots, and other organic debris. A substantial amount also comes from the roots themselves, which exude carbon-rich compounds to feed their microbial partners, particularly the mycorrhizal fungi we discussed earlier. When plants and animals die, their organic remains become food for the microscopic metropolis dwelling beneath the surface. Bacteria, fungi, and other decomposers begin the slow, deliberate process of breaking down this complex organic matter. As these microorganisms consume and transform the organic material, they respire, just like humans, releasing carbon dioxide back into the atmosphere. This is the exhalation part of soil's breathing. However, a crucial portion of that carbon is not immediately returned to the air. Instead, it is incorporated into the bodies of the microbes themselves, or it is transformed into more stable, recalcitrant forms of organic matter, collectively known as humus. Humus is highly resistant to further decomposition and can remain in the soil for decades, centuries, or even millennia. It is this stable soil organic carbon, or ESOC, that represents a vast, long-term carbon sink. To put this in perspective, the upper meter of soil globally holds an estimated 1,500 to 2,400 gigatons of carbon, a staggering amount that exceeds the carbon stored in the entire atmosphere, around 800 gigatons, and all terrestrial vegetation combined, about 550 gigatons. This makes soil the largest terrestrial carbon reservoir on Earth. The capacity of soil to sequester carbon is not uniform across the globe. Factors like climate, vegetation type, and soil texture play a crucial role. For instance, soils rich in clay minerals tend to protect organic matter more effectively because clay particles can bind to organic molecules, shielding them from microbial attack. Peatlands, found in cold, waterlogged environments like the boreal forests, are another prime example of massive carbon storage. Here, low oxygen levels slow decomposition to a crawl, allowing vast quantities of partially decayed organic matter to accumulate over thousands of years, holding gigatons of carbon. However, draining or disturbing these peatlands can rapidly release this stored carbon, turning them from carbon sinks into significant sources of greenhouse gases. The balance of carbon sequestration and release is incredibly delicate. When soil is disturbed, for example, through intensive tillage in agriculture, the physical disruption breaks apart soil aggregates. These aggregates act like tiny fortresses, protecting organic matter within their structure. Once broken, the exposed organic matter becomes readily accessible to microbes, leading to a rapid burst of decomposition and a significant release of carbon dioxide into the atmosphere. This is why practices like no-till farming and cover cropping are so vital. They minimize disturbance, allow soil to rebuild its structure, and enhance its ability to hold onto carbon, effectively slowing down Earth's exhalation of greenhouse gases. The speed at which humans have altered this ancient balance in mere decades stands in stark contrast to the geological timescales over which soil carbon typically accumulates. Beyond its critical role in regulating atmospheric carbon, soil is also an indispensable player in the global water cycle. Picture a heavy rain shower cascading onto a landscape. What happens to that water? If the soil is healthy and well structured, it acts like a giant thirsty sponge. Its intricate network of pores, created by the burrowing of earthworms and other invertebrates, and the binding action of fungal hyphae and organic matter, allows water to infiltrate efficiently rather than simply running off the surface. This infiltration is crucial for recharging groundwater aquifers. the vast subterranean reservoirs that supply much of our drinking water and sustain rivers and lakes during dry periods. As water percolates downwards through the soil profile, it undergoes a remarkable transformation. Soil acts as a natural filter and purifier, cleaning the water of contaminants. This purification happens on multiple levels. Physically, the soil particles themselves, sand, silt, and clay, act as a sieve, trapping larger suspended solids like sediment, pathogens, and debris. Chemically, the charged surfaces of clay minerals and organic matter can absorb or bind to dissolved pollutants such as heavy metals, pesticides, and excess nutrients like nitrates. This process essentially holds these harmful substances in place, preventing them from contaminating deeper groundwater or flowing into surface waterways. Biologically, the soil's microscopic metropolis plays an equally vital role in water purification. Many soil bacteria and fungi are adept at breaking down organic pollutants, including complex pesticides and industrial chemicals, transforming them into less harmful compounds or even converting them into plant-available nutrients. This bioremediation capacity of soil is a powerful, self-renewing service provided by nature. Imagine a complex, multi-stage water treatment plant operating tirelessly, silently, and without human intervention, ensuring that the water flowing into our wells and streams is clean and safe. This is precisely what healthy soil accomplishes, providing clean fresh water that is essential for all life. The water-holding capacity of soil is another critical function. Organic matter, in particular, can hold many times its weight in water, acting as a reservoir that slowly releases moisture to plant roots during dry spells. This is why soils rich in organic matter are more resilient to drought and support more robust plant growth, even with fluctuating rainfall patterns. Without this natural storage and filtration system, our rivers would be choked with sediment, our groundwater contaminated, and our landscapes prone to devastating floods followed by severe droughts. The very availability of clean, fresh water, a fundamental human right and ecological necessity, is inextricably linked to the health of the soil beneath our feet. Ultimately, soil forms the indispensable foundation for nearly all terrestrial ecosystems. Its health directly impacts biodiversity, climate regulation, and food security for human populations. Consider biodiversity, the sheer variety of life on Earth. While we often focus on charismatic megafauna or vibrant rainforests, the true engine of terrestrial biodiversity begins in the soil. Soil provides the physical habitat for countless organisms, from microscopic bacteria to burrowing mammals. A healthy, diverse soil microbiome supports a diverse community of plants, which in turn supports a diverse array of animals above ground. Each plant species has evolved unique relationships with specific soil microbes and fungal networks, and the decline of soil health can lead to the loss of these intricate ecological partnerships, unraveling the web of life from the bottom up. In terms of climate regulation, we've already explored soil's profound influence on the carbon cycle. Its capacity to sequester carbon acts as a natural brake on rising atmospheric carbon dioxide levels. But soil also influences climate in other ways. Its moisture content affects local temperatures through evaporative cooling. The presence of healthy vegetation, supported by a healthy soil, can influence regional weather patterns by increasing transpiration and cloud formation. Conversely, degraded soils, stripped of vegetation, contribute to desertification, increased dust storms, and localized heat islands, exacerbating the challenges of a changing climate. Perhaps the most direct and tangible impact of soil on human populations is its role in food security. Nearly all the food we eat, whether directly from plants or indirectly from animals that consume plants, originates in the soil. Healthy soil provides the essential nutrients, nitrogen, phosphorus, potassium, and a host of micronutrients that plants need to grow. It also stores the water that sustains crops through their life cycle and anchors them physically. When soil degrades through erosion, nutrient depletion, or compaction, its ability to produce food diminishes. We have seen this play out tragically throughout history. Ancient civilizations in the Fertile Crescent, for instance, once flourished on rich alluvial soils, but unsustainable agricultural practices eventually led to salinization and degradation, contributing to their decline. More recently, the Dust Bowl of the 1930s in the American Great Plains demonstrated how years of intensive tillage and drought, coupled with a lack of understanding of soil ecology, could turn once fertile farmlands into barren, windswept deserts, causing immense human suffering and displacement. Conversely, regions that have adopted sustainable soil management practices, such as regenerative agriculture, are demonstrating how healthy soil can enhance food production, improve crop resilience to extreme weather, and even increase the nutritional density of food. The ability of soil to support life is not an abstract concept. It is the very bedrock of human civilization. Without healthy soil, there is no food, no clean water, and ultimately, no sustainable future for our growing global population. So, when we consider soil, we are not simply looking at inert dirt. We are observing Earth's complex, living skin, performing an intricate, indispensable dance of life and death, absorption and release. It is a slow, deliberate system, constantly cycling the planet's most vital elements, regulating its climate, and underpinning the very existence of every terrestrial ecosystem. Its silent, continuous work is the invisible engine that powers our world, making it habitable and productive. To disregard its health is to jeopardize our own. We often find ourselves enchanted by the vastness of the cosmos, pondering the existence of life on distant exoplanets, or marveling at the intricate dance of celestial bodies. Yet, in our quest for grand mysteries, we frequently overlook an equally profound and infinitely more accessible wonder right beneath our feet. For many, the ground we walk on is simply dirt, an inert, uniform, and lifeless medium. This perception, however, is perhaps one of the most significant misconceptions we hold about our planet, a misunderstanding that profoundly impacts our relationship with the very foundation of terrestrial life. Imagine for a moment a child playing in a garden, scooping up a handful of earth. To them, it might be a gritty, brown substance, perhaps a little damp, perfect for building a small mound or making mud pies. An adult, too, might see it as merely a substrate for plants, something to be tilled, fertilized, and then ignored until the next planting season. This view, deeply ingrained in our collective consciousness, strips soil of its true identity. It reduces a dynamic, living, breathing ecosystem to a mere inanimate material, a backdrop rather than a protagonist in the unfolding drama of life. Yet, as we have begun to uncover, soil is anything but inert. It is a bustling subterranean metropolis, a complex web of interactions, a living skin that literally breathes and sustains our world. To call soil just dirt is akin to calling a human being just a bag of chemicals. While technically true at a fundamental level, it misses the entire essence of what makes something alive, functional, and interconnected. Healthy soil, far from being uniform, is a heterogeneous mixture of mineral particles, sand, silt, and clay, bound together by organic matter and permeated by air and water. This intricate architecture creates a diverse array of microhabitats, each hosting its own specialized community of organisms. It is a constantly transforming entity, shaped by geological forces, climatic conditions, and the ceaseless activity of its countless inhabitants. Day and night, unseen chemical reactions unfold, biological interactions drive nutrient cycles, and physical transformations reshape its very fabric. The subtle, earthy scent after a rain, often attributed to petrichor, is not just wet dust. It is partly the metabolic byproducts of soil bacteria, a tangible sign of its vibrant, unseen life. Furthermore, viewing soil as merely an inert growing medium fundamentally misunderstands its partnership with plants. It is not simply a physical anchor providing stability. Instead, soil acts as a vital, active partner, an indispensable lifeline for nearly all terrestrial vegetation. It is the primary provider of essential nutrients, meticulously cycling elements like nitrogen, phosphorus, and potassium from organic matter and mineral reserves into forms that plants can absorb. It regulates water availability, acting like a giant sponge, absorbing rainfall, filtering it, and then slowly releasing it to plant roots, preventing both drought and waterlogging. Moreover, the soil microbiome actively defends plants against pathogens, synthesizes growth-promoting hormones, and even facilitates communication between different plant species through intricate fungal networks, as we explored earlier. Without this active living partnership, plants would struggle to thrive, and terrestrial ecosystems as we know them would collapse. The subtle hum of life beneath our feet is not just a poetic notion. It is a fundamental truth, a testament to the continuous quiet work of billions of organisms creating the conditions for life above ground. Recognizing these fundamental misconceptions sets the stage for appreciating the cutting edge of scientific inquiry, where researchers are peeling back the layers of soil's complexity with unprecedented tools. One of the most dynamic and rapidly evolving fields is the study of the soil microbiome. For centuries, our understanding of soil microorganisms was limited to what could be cultured in a laboratory, a tiny fraction of the actual diversity. Today, however, advancements in molecular biology, particularly DNA sequencing and metagenomics, have revolutionized this field. Scientists can now extract all the DNA from a soil sample and identify thousands of different species of bacteria, archaea, fungi, protozoa, and viruses that inhabit it, many of which have never been seen alive. This has revealed an astonishing biodiversity, with a single gram of soil about the weight of a paperclip, potentially containing billions of individual organisms and tens of thousands of different species. Researchers are working to understand not just who is there, but what they do. Through techniques like stable isotope probing, where specific molecules are tagged with heavy isotopes, scientists can track which microbes are consuming particular substances and what metabolic pathways they are employing. This allows them to pinpoint the functional roles of different microbial groups in processes like nutrient cycling, organic matter decomposition, and even the detoxification of pollutants. The soil microbiome, it turns out, is like an incredibly complex, redundant, and robust factory, with multiple teams of workers capable of performing similar tasks, ensuring the system continues to function even if some members are lost. However, much of this remains a black box. We are only just beginning to decipher the intricate language and interactions within this hidden world. Understanding these microbial communities is crucial, as they are the primary drivers of soil health, directly influencing plant growth, disease suppression, and the overall resilience of ecosystems. Another critical area of current research focuses on soil's resilience to climate change. As global temperatures rise and weather patterns become more erratic, understanding how soil ecosystems respond and how they can help mitigate these changes is paramount. Scientists are studying how increased temperatures accelerate the decomposition of soil organic matter, potentially releasing vast amounts of stored carbon back into the atmosphere, creating a dangerous feedback loop. They are also investigating the impact of altered precipitation patterns, from prolonged droughts that desiccate microbial communities and halt biological activity, to intense floods that lead to severe erosion and nutrient loss. The thawing of permafrost in Arctic regions, for example, is a major concern, as it could unlock ancient carbon stores, releasing potent greenhouse gases like methane and carbon dioxide on a scale that could dramatically accelerate global warming. Conversely, research is also exploring how healthy soil can enhance resilience. Soils rich in organic matter act as natural sponges, improving water infiltration during heavy rains and increasing water retention during dry spells, making ecosystems more tolerant to extreme weather events. Scientists are investigating how diverse microbial communities can adapt to environmental shifts, maintaining critical functions even under stress. The ability of soil to sequester carbon is also a key area. By enhancing carbon storage through improved land management, soils can become a powerful tool in climate change mitigation. This involves complex models and field experiments to quantify how much carbon can be stored, at what rate, and for how long, often examining the long-term stability of different forms of soil carbon. This leads directly to the growing interest in regenerative agriculture, a set of holistic farming practices designed to improve soil health, biodiversity, and ecosystem services. Unlike conventional agriculture, which often relies on heavy tillage, synthetic fertilizers, and pesticides, regenerative practices aim to work with natural processes. Key components include minimizing soil disturbance, no-till or reduced tillage, maximizing crop diversity, diverse rotations and cover cropping, keeping the soil covered with living roots for as long as possible, integrating livestock, and minimizing synthetic inputs. Researchers are actively studying the efficacy of these practices across different agro-ecosystems. For instance, long-term field trials at institutions like the Rodale Institute in Pennsylvania have demonstrated that regenerative organic systems can build soil organic matter over time, increase water infiltration rates, and often match or exceed conventional yields, especially during droughts, all while reducing energy inputs and greenhouse gas emissions. Similar successes have been observed on farms like Gabe Brown's in North Dakota, where a commitment to regenerative principles has transformed degraded land into highly productive, resilient ecosystems. However, the scientific community is also engaged in robust debates surrounding these very topics. One such debate revolves around what constitutes optimal soil management. While regenerative practices show great promise, there is no single, universally applicable blueprint. Soil types vary dramatically from sandy loams to heavy clays. Climates range from arid to humid, and agricultural systems encompass everything from row crops to perennial orchards. What works best in one context might not be ideal in another. For example, the extent of tillage reduction is often debated. Is absolute no-till always superior? Or can strategic minimal tillage sometimes be beneficial for specific soil types or pest issues. Similarly, the role of synthetic inputs remains a contentious point. While regenerative agriculture aims to reduce or eliminate them, some argue that judicious, targeted use of certain inputs might be necessary in specific situations to maintain productivity, especially during the transition phase from conventional to regenerative systems. Researchers are working to develop adaptive management strategies, recognizing that optimal is a dynamic and context-dependent concept, requiring continuous monitoring and adjustment. Another significant area of discussion centers on carbon sequestration metrics. While it's widely accepted that healthy soils store carbon, precisely quantifying this storage and its permanence is a complex challenge. How do we accurately measure changes in soil carbon over time, considering the vast spatial variability of soil properties, the depth to which carbon is stored, and the different forms it takes, from labile organic matter to more stable mineral-associated carbon? Current methodologies, including soil sampling, remote sensing, and ecosystem modeling, each have their limitations and uncertainties. For instance, soil sampling can be labor-intensive and expensive, and results can vary widely depending on sampling depth and frequency. Remote sensing offers broad coverage, but often lacks the resolution to detect subtle changes in soil carbon. The debate intensifies when discussing carbon credits and markets. How can we ensure that sequestered carbon is truly additional, meaning it wouldn't have happened without the intervention, permanent, not re-released into the atmosphere after a few years, and verifiable? What are the appropriate baselines for measuring carbon gains? These questions are critical for developing credible carbon markets that incentivize farmers to adopt climate-friendly practices. Scientists are striving to refine measurement techniques, develop more robust models, and establish standardized protocols to ensure accurate and reliable carbon accounting, which is essential for both environmental integrity and economic viability. Finally, the precise quantification of soil health benefits presents its own set of challenges and ongoing debates. While the qualitative benefits of healthy soil, such as improved water quality, enhanced biodiversity, and increased crop resilience, are widely acknowledged, putting exact numbers on these advantages can be difficult. How do we assign an economic value to the reduction in nutrient runoff that prevents algal blooms in waterways? What is the monetary benefit of a more diverse soil microbiome that suppresses plant diseases, reducing the need for fungicides? How much of a yield increase can be directly attributed to improved soil health versus other factors like favorable weather or genetic improvements in crops? Researchers are actively working to develop standardized indicators for soil health, moving beyond simple organic matter content to include metrics like aggregate stability, microbial biomass and respiration, nutrient cycling rates, and water infiltration capacity. The challenge lies in identifying a core set of indicators that are reliable, cost-effective to measure, and broadly applicable across different soil types and agricultural systems. Translating complex scientific findings into practical, easily understandable metrics for farmers, policymakers, and consumers is a crucial step in promoting wider adoption of soil-friendly practices. These debates are not signs of scientific uncertainty about the importance of soil, but rather a healthy and necessary part of refining our understanding and developing the most effective, evidence-based strategies for its management. They underscore the incredible complexity of soil and the ongoing journey of discovery that continues to unfold beneath our feet. Our journey beneath the surface has revealed a world far richer and more complex than most of us ever imagined. from the teeming microbial metropolises in a single handful of earth to the vast unseen fungal networks that connect entire forests, we have learned that soil is anything but inert. It is a living, breathing entity, a silent partner in nearly every process that sustains life on our planet. Now, as we draw our exploration to a close, it is time to connect these profound scientific discoveries to our everyday lives, to understand the immediate and long-term implications of soil health for human well-being and the stability of the entire Earth system. The secret life of soil is not just a fascinating academic topic, it is the very foundation of our future. Perhaps the most direct and undeniable link between soil health and human survival is food security. Every single meal we consume, every grain of rice, every vegetable, every piece of fruit, every animal product, traces its origins back to the soil. Healthy soil is, quite literally, the engine of our food systems. It provides the essential nutrients that plants need to grow, acting as a colossal slow-release fertilizer. Microorganisms, the billions of bacteria and fungi we discussed, are the tireless workers in this system, breaking down organic matter and converting complex compounds into forms that plant roots can absorb. They cycle nitrogen, phosphorus, and potassium, the macronutrients vital for plant growth, along with a host of essential micronutrients like iron, zinc, and manganese. When soil is vibrant and teeming with life, these nutrients are abundant and readily available, leading to robust, healthy crops that are more resilient to pests and diseases. Beyond nutrient provision, healthy soil plays an indispensable role in water management for agriculture. Imagine soil as a colossal sponge, capable of absorbing and holding vast quantities of water. This remarkable capacity is largely due to its organic matter content and stable aggregate structure, created by the binding activities of fungal hyphae and bacterial exudates. When rain falls, healthy soil allows it to infiltrate deeply, rather than running off the surface. This dramatically reduces erosion, preventing the loss of precious topsoil, and ensures that water is stored for plants to access during dry spells. In contrast, degraded soil, often compacted and low in organic matter, behaves more like a paved surface. Rain hits it, washes away, taking with it valuable nutrients and soil particles, and leaving plants vulnerable to drought even after significant rainfall. This difference can be seen starkly in fields after a heavy storm. A healthy field might still have moist soil and standing crops, while a degraded field shows visible rills of erosion and parched, struggling plants. The ability of soil to regulate water directly translates to higher and more consistent crop yields, providing a stable foundation for feeding a global population that is projected to reach nearly 10 billion people by the middle of this century. Without productive soil, the challenge of feeding ourselves becomes insurmountable. Another critical service provided by healthy soil is the purification of our water. The ground beneath our feet acts as Earth's most effective natural filter. As water percolates through the layers of soil, it encounters a complex matrix of mineral particles, organic matter, and an astonishing diversity of microorganisms. This matrix physically traps sediment and larger particles, while the chemical properties of soil, such as the charged surfaces of clay and organic matter, absorb dissolved contaminants like heavy metals and pesticides. Crucially, the soil microbiome contributes a biological filtering capacity. Many bacteria and fungi are capable of breaking down harmful organic pollutants, transforming them into less toxic or even inert substances. This natural purification process ensures that the water that eventually reaches underground aquifers, from which much of our drinking water is drawn, is cleaner and safer. When soil is degraded, however, its filtering capacity diminishes. Compacted soils with poor structure allow water to bypass the filtration layers, carrying pollutants directly into groundwater. Runoff from agricultural fields, laden with excess fertilizers and pesticides, can surge directly into rivers and lakes, causing widespread contamination, leading to harmful algal blooms that deplete oxygen and harm aquatic life, and making water unsafe for human consumption. Our access to clean, potable water is thus inextricably linked to the health of the soil through which it must pass. The silent work of soil also plays a monumental, though often overlooked, role in climate change mitigation. We have previously discussed soil as the largest terrestrial carbon reservoir, holding more carbon than the atmosphere and all plants combined. This carbon is stored primarily in the form of organic matter, the decomposed remains of plants and animals, which is stabilized by microbial activity and bound within soil aggregates. When plants photosynthesize, they draw carbon dioxide from the atmosphere and convert it into sugars. A significant portion of these sugars is then transferred to the roots and exuded into the soil, feeding the microbial community and contributing to the formation of stable soil organic carbon. This process, known as carbon sequestration, is a natural and highly effective way to remove greenhouse gases from the atmosphere. However, conventional agricultural practices, particularly intensive tillage, disrupt this delicate balance. Tillage breaks up soil aggregates, exposing previously protected organic matter to oxygen and accelerating its decomposition by microbes, which then release carbon dioxide back into the atmosphere. Erosion also plays a destructive role, washing away carbon-rich topsoil. By adopting regenerative agricultural practices such as no-till farming, cover cropping, and diverse crop rotations, we can reverse this trend. These methods minimize soil disturbance, keep living roots in the ground year-round, and increase the input of organic matter, effectively turning agricultural lands into more powerful carbon sinks. Even small increases in soil organic carbon across vast agricultural landscapes can translate to significant reductions in atmospheric carbon dioxide, offering a powerful, nature-based solution to the climate crisis. Furthermore, soil health is fundamental to the preservation of biodiversity, both below and above ground. The soil itself is a biodiversity hotspot, home to a staggering array of life forms from bacteria and archaea to fungi, protozoa, nematodes, mites, insects, and earthworms. This subterranean web of life is incredibly intricate, with each organism playing a specific role in nutrient cycling, decomposition, and maintaining soil structure. When soil is healthy, it supports a diverse community of these organisms, which in turn creates a resilient and productive ecosystem. This biodiversity beneath our feet is directly linked to the biodiversity we see above ground. A diverse soil microbiome supports a wider variety of plant species, which then provide food and habitat for a greater diversity of insects, birds, and mammals. Conversely, soil degradation, often driven by monoculture farming, excessive use of synthetic fertilizers and pesticides, and habitat destruction, leads to a dramatic loss of soil biodiversity. This loss weakens the entire ecosystem, making it more vulnerable to pests, diseases, and environmental stresses. Imagine the intricate, self-sustaining forest ecosystem, its towering trees, its understory plants, its abundant wildlife. all are ultimately reliant on the health and diversity of the soil that cradles their roots. Without this vibrant foundation, the entire ecological edifice begins to crumble. To truly grasp the profound consequences of our stewardship, or lack thereof, let us consider some real-world examples. The infamous Dust Bowl of the American Great Plains in the 1930s stands as a stark historical warning. Driven by decades of aggressive ploughing that removed native prairie grasses and left the soil exposed, coupled with a severe drought, the topsoil, once a rich, dark brown, simply blew away. Gigantic dust storms, some reaching thousands of meters into the sky, swept across the landscape, darkening skies as far east as New York City. Farms were rendered unproductive, livelihoods were destroyed, and millions of people were displaced in one of the largest ecological and economic disasters in American history. This catastrophic event was a direct consequence of treating soil as an inert medium to be exploited, rather than a living system to be nurtured. Across the globe, in the Sahel region of Africa, a slower yet equally devastating form of soil degradation known as desertification is ongoing. Here, a combination of overgrazing, unsustainable farming practices, and the relentless pressures of a changing climate are gradually transforming once productive rangelands and croplands into barren desert. As vegetation cover diminishes, the soil is exposed to the harsh sun and wind, losing its organic matter, its ability to hold water, and its fertility. This leads to reduced food production, increased poverty, and mass migration, illustrating how soil degradation can unravel the social fabric of the entire regions, creating humanitarian crises of immense scale. These examples serve as powerful reminders of the long-term, devastating consequences when we fail to respect the delicate balance of soil ecosystems. However, the story is not one of inevitable decline. There are inspiring examples of successful restoration efforts that demonstrate the immense potential for healing our planet's skin. Consider the remarkable transformation of China's Lulls Plateau. For centuries, this vast region, characterized by deep, erodible low soils, suffered from extreme erosion, leading to devastating floods and widespread poverty. In the 1990s, a monumental project began, focusing on reforestation, terracing, and sustainable land management. Farmers were encouraged to adopt practices that kept soil covered and minimized disturbance. Decades later, satellite images show a dramatic change. Once barren hillsides are now green with trees and vegetation. Erosion has been significantly reduced, and the livelihoods of millions of people have improved. This project stands as a testament to what is possible when human ingenuity and sustained effort are directed towards restoring soil health on a massive scale. Closer to home, and perhaps more replicable, are the growing number of farmers worldwide who are embracing regenerative agriculture. These pioneers are rebuilding soil health on their farms by implementing practices like no-till farming, planting diverse cover crops, rotating crops strategically, and integrating livestock grazing in a holistic manner. In places like Gabe Brown's farm in North Dakota, or the thousands of farms participating in organizations like the Rodale Institute, farmers have seen their soil organic matter levels rise from less than 1% to over 6 or 8% in just a few decades. This translates to increased water infiltration, reduced need for synthetic fertilizers and pesticides, greater resilience to extreme weather events like floods and droughts, and ultimately, more profitable and sustainable operations. These examples demonstrate that healthy soil is not just an environmental imperative, but an economic one, offering a pathway to a more secure and resilient future for agriculture. Ultimately, our journey into the secret life of soil culminates in a profound realization. Soil is not merely a resource, a commodity to be exploited, or a passive medium for growth. It is a living, slow-thinking system, a complex, interconnected intelligence that underpins the very existence of terrestrial life. It remembers the history of the landscape, responding to every input, every disturbance, every act of care or neglect. Its health dictates not only the quality of our food and water, the stability of our climate, and the richness of our biodiversity, but quite literally, our own survival and future prosperity. The legacy we leave for future generations will be etched not just in stone or steel, but in the quality of the soil we bequeath to them. Will it be a depleted, degraded landscape, struggling to sustain life, or a vibrant, fertile foundation capable of nourishing a thriving world? The choice is ours, and it rests on our understanding and respect for this quiet, invisible engine of the planet. Let us not gaze only at the stars, but also look down with wonder and profound gratitude at the living legacy beneath our feet. For in the health of the soil lies the health of all.